Now we are talking about microarray, which is an important tool in estimating the gene expression. Remember guys, whenever we talk about gene expression, we mean the production of messenger RNA. So genes are expressed, that is they are transcribed, the process we call it as transcription, and then they are giving the messenger RNAs. And then these messenger RNA, they go through a process of translation where they make up their proteins. So whenever we are talking about the expression of genes, actually we are talking about the expression in terms of how many molecules of messenger RNA they are producing. So this microarray actually consists of small membranes or glass slides containing samples of many genes arranged in a regular pattern. And it has become incredibly popular since their inception in 1995. When we talk about gene expression, it involves detection of messenger RNA. This messenger RNA complement of the cell is also called as transcriptome. So how much amount of messenger RNA is present in a cell in a particular tissue at a particular time. So this is what we are doing here. This messenger RNA, if it is isolated from a cell, so it can give us an idea or a snapshot of the events occurring at that time. So how many genes are expressed and what functions that may be affected by these, the expression of these genes. So gene expression studies, they lead to methods capable of giving a snapshot of RNA expression of all genes. They can be used as diagnostic profiles. So for example, in cancers and in some other diseases or in any other environmental factors which are influencing the organism, we can get the profile or the expressions of different genes altogether. They can show how RNA level changes during development, exposure to stimuli, and cell cycle. It produces a large amount of data. So whenever huge amount of data is produced, we need really important and very skilled techniques like statistics and computational biology techniques to explore that data and grab interesting patterns out of that data. So they can help understand the functions of the whole systems since we are pulling the whole RNA from the whole cells and we are taking thousands of the genes at a time. So in this way we can get the idea of, we can talk about the system level biology. Basic principle behind microarray is that DNA can hybridize to itself, RNA can hybridize to DNA, and RNAs they can also hybridize with one another. So it's based upon the process of hybridization. If you look into the evolution of microarrays, so in the beginning we have those one gene, one experiment studies, so where you can have a gene and you want to capture its RNA, so you might use the techniques like this northern blots where you can capture that messenger RNA by maybe something like you can target the poly A tail of that messenger RNA, and in this way you can capture that. So you can design oligo-DTs, you can capture it with some other RNA, or people used to have DNA also to capture that messenger RNA. So basically the principle was this hybridization. And then RNase assays, so in which the enzymes that cleave RNA, they were used. So these RNases, etc., they cut this RNA into small chunks or pieces. So you can take the RNA and then you can let it hybridize with the DNA. And one way is you can use these RNAs, and in which you can remove these RNAs, and then you can capture what is the parent gene in which it was bounded. Real-time PCRs, those kind of techniques, they are on real-time techniques where the messenger RNA which is produced in the cells and then it's converted into the cDNA, and then that DNA is amplified. So in this way you can capture, or you can amplify its quantity, and then you can capture its expression. So those genes which are expressed at higher amount, the probability is you will amplify them more. So in this way it can gives you an idea about their relative expression with one another. Then came another technique called as macroarrays. So in macroarrays, they use nylon membranes, and then they also use 32p or phosphorus labeled probes. So we can have RNA and we can convert it into, or we can let it attached with the cDNA, where you have those 32 P's. So in this way, they hybridize with one another. So wherever you have those probes, which are cDNA probes, which are labeled, wherever they go and bind with the RNA. So in this way, you can capture the RNA and you can have an idea about their relative intensity, or you can have their sequences. Then came the slide-based arrays. So the probe DNA is affixed directly into the surface of a glass slide, and the probe then can range from a medium length oligos. So you might have oligonucleotides like up to 60 nucleotides to an entire cDNA clone or larger. You might have over 1000 nucleotides in length, these cDNA arrays, and you can also have cDNA libraries to be used. So in general, this microarray can be divided into two basically steps. Number one is RNA extraction, and the second one is cDNA formation as well as labeling with the dyes. So we have, for example, a tissue which is 1 or A, and then we have a B. One of them is green, one of them is orange in color. So we capture their RNAs. So in this way, those RNAs we normally capture by using their tails. So these tails of those messenger RNAs are polyadenylated. We call them as poly-A tails. We capture them and then we convert them into the cDNA. So when we convert them into the cDNAs, these tails then they become poly-Ts, and we take one sample, the green one, and we use, for example, the green is just a cell color which is just over here, is just for an elaboration. So we take this one and we put a dye CY3. So CY3 gives it a green color, and then CY5 is used, is transferred onto those messenger RNA. So that is actually a dye which gives a red color. So whenever we pass laser through them, we will get a red and a green color pattern. So where we have those hybridizations, cross hybridization, or where we have the spots where we have both these species, we can expect a mixture of colors like yellow, etc. We call this first sample as A, and we call the second sample as B. Maybe a tissue type A and a tissue type B. Maybe we have a control versus treated, different kind of situations we can have it. Then we take these probes and we let those to be attached on these slides. So we have those probes attached and we have the concentration of messenger RNA in a mixture. So we put it there. So we will observe different cross hybridizations and we will observe different color patterns. So for example, in a spot, you have the messenger RNAs. Here we magnify an area. So we have green color spot. So here we can see that more in A. So mostly the RNA species which are here in this region, they are actually more A's, type A tissue is present, since we see the color is green. We have orange or red. So here we see that it is more in B. And we do have yellow colors where we have equal proportion of A and B. And in between we have different shades. So more stronger in A, we will get the green. So we have those samples which are labeled. And then we have the probes which are there to capture them. So in this way, when we put them on the slide, so we have a relative abundance of different species, maybe A, maybe B, maybe both of them present in a spot. Microarray can be used in gene discovery. We can have the idea about tissue profiles, the expression profiles within different tissues. We can capture time course expression profiles or data where we capture their messenger RNAs at different time intervals, that may be some time intervals within a 24-hour cycle, or maybe spaced by some days or weeks, where we can also have altered genetic backgrounds, so we can also capture their expressions, and comparing tissues and genotypes. So we see that microarrays are used to investigate the gene expression, and microarrays are based upon the principle of hybridization.